In each of your cells, DNA is transcribed into RNA molecules to create proteins that are critical for life. Delivering specially designed RNA molecules directly into cells has emerged as a potential way to vaccinate against or treat cancer and infectious diseases. However, creating methods to deliver RNA efficiently into cells has proven to be very challenging. But what if there was a tool that could be strategically implanted into cells to manufacture RNA on demand? The Chou lab at the University of Toronto aims to address this need with the RNA Nanofactory. The factory is a nano device built out of DNA. It is studded with functional molecular units. The modular design of the factory allows it to be programmed to execute unique tasks that can dramatically alter cells and their environment. Could the nanofactory produce functional RNA? Could this nanodevice change the way we receive treatments or vaccines? Let's find out. The RNA Nanofactory is constructed using DNA origami and comprises hundreds of short DNA strands that self-assemble and hybridize to different regions of a longer DNA strand, folding it into a user-defined 3D shape. The factory's design makes use of 4 such origami objects, 2 outer peripheral barrel monomers, and 2 connected central barrels. Each barrel is equipped with addressable sites, which allow for additional molecular components to be arranged on the surface of the barrel. The addressable sites are single-stranded DNA extensions, and each barrel monomer can be programmed with 48 handles on the interior and 60 handles on the exterior. The unique design of the RNA Nanofactory includes 12 RNA production units and 48 RNA processing units within the lumen of each central and peripheral barrel, respectively. The RNA production units are designed for rolling circle transcription and include a circular DNA template, T7 RNA polymerase, and a DNA leash. The circular DNA leash is bound to one of the addressable sites on the central barrel and runs through the center of the DNA template, forming an interlocking loop known as a catenane with the DNA template. The catenane allows the double-stranded DNA template to freely rotate while still being tethered to the nanofactory lumen during rolling circle transcription. T7 RNA polymerase is used to transcribe the DNA template and is modified with a single-stranded DNA oligonucleotide. It is complexed with the circular DNA template through DNA hybridization with the mechanically locked circular DNA leash. The RNA processing units work in conjunction with the production units to manufacture mature RNA. The RNA processing unit used in the experiments is a mutated endoribonuclease Cas6e, known to have multi-turnover ribonuclease activity. Each Cas6e is covalently attached to a single-stranded DNA oligonucleotide, which hybridizes to an addressable site within the peripheral barrels. Following rolling circle transcription, long RNA concatemers are generated by the RNA production units. These nascent RNA transcripts must then run the gauntlet of RNA processing units before exiting the polar ends of the nanofactory. Here they encounter the Cas6e enzymes, which cleave them into functional monomeric RNA transcripts. The RNA Extruding Nanofactory establishes a platform for RNA production in vitro and demonstrates the potential of DNA nanodevices for integrated production and processing of RNA. It highlights the potential of DNA-based nanomachines to manufacture tailor-made molecular therapies for cancer and other infectious diseases. The Chou lab is continuing to advance the development of the RNA Nanofactory with the goal of integrating the nanodevice into intracellular environments. To learn more about Chou Lab's work in DNA biotechnology and the RNA Extruding Nanofactory, visit choulab.org, CHOULAB.org.